restriction enzymes. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease into whose functioning different non-specific DNA nucleotide sequence was isolated and characterized. 5 years later, it was found that the hint to always cut DNA molecules at a particular point by recognizing a specific sequence of six bases where this specific base sequence was none other recognition sequence for Hindu. Besides Hindu, today we know more than 900 restricted enzymes that have been isolated from over 230 strands of bacteria, each of which recognize different recognition sequence. The convention for naming these enzymes is first letter of the name comes from the genus and the second two letters come from the species of the prokaryotic cell from which they were isolated. Example, E. coli comes from Escherichia coli RY30. In the E. coli, the letter R is derived from the name of the strain. Roman numbers following the names indicate the order in which enzymes were isolated from that strain of bacteria. Restriction enzymes belong to a larger class of enzymes called enuclease. These are of two kinds, exonuclease and endonuclease. Exonuclease remove nucleotide from the ends of the DNA, where endonuclease makes cut at specific position within the DNA. Each restriction endonuclease is functioned by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at a specific point in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. These are group of letters that form the same word when read both forward and backward. Malayalam as against word palindromic, where the same word is read in both directions. The palindrome in DNA is a sequence of base pair that reads same on the two strands, where orientation of reading is kept in the same. For example, the following sequences read the same on the two strands in 5:3 direction. This is also true if reads in 3:5 direction. GAATTC CTTAAG. Restriction enzyme cut the strands of DNA a little away from the center of the palindromic sites, but between the same two bases on the opposite strands. This leaves single stranded portion at the end. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bond with their complementary cut counterpart. This thickness of ends facilitates the action of the enzyme DNA ligase.